Okay, we're starting on page 52. Forming a two-headed planarian. Any such field must be able to stimulate cells to switch various genes on and off, that is, to change their specialization. A large body of research on embryonic development has identified various chemical inducers, compounds that stimulate neighboring cells to differentiate in a certain fashion, producing the next type of cells needed. But these substances act only on the basis of simple diffusion. Nothing in the way they operate can account for the way the process is controlled to express the overall pattern. Another classic experiment helps clarify the problem. A salamander's hand can be amputated and the wrist stump sewn to its body. The wrist grows into the body and nerves and blood vessels link up through the new connection. The limb now makes a U shape connected to the body at both ends. It's then amputated at the shoulder to make a reversed limb attached to the body at the wrist and ending with the shoulder joint. The limb then regenerates as though it had simply been cut off at the shoulder. The resulting limb looks like this. From the body sprouts the original wrist, forearm, elbow, upper arm, and shoulder, followed by new upper arm, elbow, forearm, wrist, and hand. Why doesn't the regenerate conform to the sequence already established in the limb instead of following as closely as possible to the body's pattern as a whole? Again, what is the control factor? Information, and a monumental amount of it, is clearly passed from the body to the blastema. Our best method of information processing at present is the digital computer, which deals with bits of data, signals that in essence say yes or no, 1 or 0. The number of such bits needed to fully characterize the salamander forelimb is incalculable, exceeding the capacity of all known computers operating in unison. The question of how this information is transferred is one of the hardest problems ever tackled by scientists. And when we fully know the answer, we'll understand not only regeneration, but the entire process of growth from egg to adult. For now, we had best, as biologists themselves have done, skip this problem and return to it after addressing some slightly easier ones. It seems reasonable that understanding what comes out of the blastema would be easier if we understood what goes into it. So the other major questions about regeneration have always been, what stimulates the blastema to form? And where do its cells come from? The idea that dedifferentiation was impossible led to the related belief that all regeneration had to be the work of neoblast or reserve cells left over from the embryo and warehoused throughout the body in primitive, unspecialized state. Some biological bell supposedly called them to migrate to the stump and form the blastema. There's evidence for such cells in hydras and flatworms, although it's now doubtful that they fully count for regeneration in these animals. However, no one ever found any in a salamander. In fact, as long ago as the 1930s, there was nearly conclusive evidence that they did not exist. Nevertheless, any anti-dedifferentiation dogma and the reserve cell theory were defended fanatically by West in particular, so that many unconvincing experiments were interpreted to prove that reserve cells formed the blastema. When I started out, it was very dangerous for one's career even to suggest that mature cells might create the blastema by dedifferentiating, because it was so hard to imagine how blastema could arise without dedifferentiation. The idea later developed that perhaps cells could partially dedifferentiate. In other words, perhaps muscle cells could become cells that looked primitive and completely unspecialized, but that would then take up their previous lives as mature muscle cells after the period of amnesia and the blastema. To fit the square peg into the round hole, many researchers did a lot of useless work, laboriously laboriously counting cell divisions to try to show that the muscle cells in a stump made enough new muscle cells to supply the regenerate. The embarrassing blastema, enigmatic and completely undifferentiated, was still there. We now know that at least some types of cells can revert completely to the primitive state, and that such despecialization is the major, probably the only way a blastema forms in complex animals like a salamander. The other major question about the blastema's origin is what triggers it? The best candidates for a carrier of the stimulus are the nerves. In complex multicellular animals, there's no regeneration without nerve tissue. Back in 1823, the English amateur Tweedy John Todd found that if the nerves into a salamander's leg were cut when the amputation was made, the limb wouldn't regrow. In fact, the stump itself shriveled up and disappeared. However, Todd got normal regeneration when he gave the nerves time to reconnect before severing the leg. Science wasn't ready to make anything of this observation, but many experiments since have confirmed it. Over a century later, Italian biologist Piero Locatelli showed that an extra leg would grow if a nerve was rerouted so that it ended near an intact leg. She cut the large sciatic nerve partway down the salamander's hind leg, leaving it attached to the spinal column and carefully threading it up under the skin so that it ends touched the skin near one of the forelegs. An extra foreleg sprouted there when she placed the nerve and end near a hind leg. An extra hind leg grew. It didn't matter where the nerve was supposed to be, the kind of extra structure depended on the target area. This indicated that some sort of energy from the nerves was adapted by local conditions that determined the pattern of what grew back. Soon afterward, other researchers found that when they sewed full thickness skin grafts over the stumps of amputated salamanders' legs, the dermis or inner layer of the skin acted as a barrier between the apical cap and an essential something in the leg, thereby preventing regeneration. Even a tiny gap in the barrier, however, was enough to allow regrowth. In the early 1940s, this discovery led S. Merrill Rose, then a young anatomy instructor at Smith College, to surmise that the rapid formation of full thickness skin over the stumps of adult frog legs might be what prevented them from regenerating. Rose tried dipping the wounds in saturated salt solution several times a day to prevent the dermis from growing over the stump. It worked. Most of the frogs whose forelimbs he'd amputated between the elbow and wrist replaced some of what they'd lost. Several regrew well-formed wrist joints, and a few even began to produce one new finger. Even though the replacements were incomplete, this was a tremendously important breakthrough, the first time any regeneration had been artificially induced in an animal normally lacking the ability. However, the dermis did grow over the stump, so that the experiment worked by some means Rose hadn't expected. Later, other investigators showed that in normal regeneration, the apical cap minus the dermis was important because regrowing nerve fibers made unique connections with the epidermal cells in the first stage of the process, before the blastema appeared. These connections are collectively called the neuroepidermal junction, or NEJ. In a series of detailed experiments, Charles Thornton of Michigan State University cut the nerves to salamander legs at various times before amputating the legs, then followed the progress of regrowing nerves. Regeneration began only after the nerves had reached the epidermis, and it could be prevented by any barrier separating the two, or started by any breach in the barrier. By 1954, Thornton had proved that the epidermal junction was the one pivotal step that must occur before the blastema could form and regeneration begin. Shortly thereafter, Elizabeth T. Hay, an anatomist then working at Cornell University Medical College in New York, studied the neuroepidermal junction. Then an electron microscope, she found that as each nerve fiber bundle reached at the end of the stump, it broke up and each fiber went its separate way, snaking into the epidermis, which might be 5 to 20 cells thick. Each nerve fiber formed a tiny bulb as its tip, which was placed against the epidermal cell's membrane, nestling into a little pocket there. The arrangement much like a synapse, although the microscopic structure wasn't as highly developed as in such long-term connections. The junction was only a bridge, however. The important question was, what traffic crossed it? In 1946, Lev Vladimirovich Palechev, a young Russian biologist then working in London, concluded a long series of experiments in which he'd induced partial regeneration in adult frogs. The same success Rose had by pricking their limb stumps with a needle every day. Polizev then found that a wide variety of irritants produced the same effect, although none of them worked in mammals. His experiments indicated that making the injury worse could make regeneration better, and showed that rose salt in the wound procedure worked by irritation rather than by preventing dermis growth. Next, the part that the nerve tissue played was clarified considerable by Marcus Singer in a brilliant series of experiments at Harvard Medical School from the mid-40s to the mid-1950s. Singer first confirmed Todd's long-forgotten work by cutting the nerves in his salamander legs at various stages of regrowth, proving that the nerves were needed only in the first week until the blastema was fully formed and the information transferred. After that, regeneration proceeded even if the nerves were cut. Recent research had found that a salamander could replace its leg if all the motor nerves were cut, but not without the sensory nerves. Many assumed then that the regrowth factor was related only to sensory nerves, but Singer was uneasy over this conclusion. The problem stated in advance that one or another nerve component is all important for regeneration, Italics added. Several facts didn't fit, however. Not only did the blastema fail to form when all nerves were cut, it didn't begin to form even if a substantial number, but still a minority, remained. Also, a salamander's leg would regrow with only motor nerves if extra motor nerves from the belly were redirected into a stump. In addition, zoologists had found that the sensory nerve contained more fibers than the motor nerve. Singer counted for himself. In the thigh or upper arm, sensory fibers outnumbered motor by 4 to 1. The ratio was even larger at the periphery. Then he cut them in various combinations in a long series of experiments. Regeneration worked as long as the leg had about 1/4 to 1/3 of its normal nerve supply, no matter what combination. There seemed to be a threshold number of neurons, nerve cells, needed for regrowth. But it wasn't that simple. The limbs of Xenopus, a South American frog unique in its ability to regenerate during adult life, have nerve fibers numbering well under the threshold. So Singer started measuring neuron size and found that Xenopus had much bigger nerves than non-regenerating frogs. Another series of experiments verified the link. A critical mass, about 30% of the normal nerve tissue, must be intact for regeneration to ensue. This finding made it pretty certain that whatever it was the nerves delivered didn't come from their own function of transmitting information by nerve impulses. If nerve impulses had been involved, regeneration should have faded away gradually with greater and greater flaws as nerves were cut, instead of stopping abruptly when the minimum amount was no longer remain. Singer's discovery also provided a basic explanation for the decline of regeneration with increasing evolutionary complexity. The ratio between body mass and total nerve tissue is about the same in most animals, but more and more nerve became concentrated in the brain, a process called encephalization. As animals became more complex, this diminished the amount of nerve fiber available for stimulating regeneration in peripheral parts, often below the critical level. In the early 1950s, Singer applied what he'd learned to the non-regenerating adult bullfrog. Using Locatelli's method, he dissected the sciatic nerve out of the hind leg, leaving it attached to the spinal cord, and directed it under the skin to the foreleg amputation stump. In 2 or 3 weeks, blastemas had formed and the cut legs were restored to about the same degree as in Rose's and Polychev's experiments. By 1954, Singer was ready to look for a regrowth-inducing chemical that was presumed to be coming from the nerves. The most promising possibility was the neurotransmitter acetylcholine, one of several compounds known to relay nerve impulses across synapses. The nerves secreted acetylcholine more abundantly than normal during blastema formation, just when nerve supply was crucial, and its production fell back to normal when regrowth was well underway. Singer had studied previous failures with acetylcholine, in which experiments had rubbed it on the stump or injected it into the blastema. He thought these methods were too artificial, so he invented a microinfusion apparatus to release tiny amounts of acetylcholine continually, just as the nerves did. He used a clock motor to drip the hormone slowly through a needle just to the shoulder of this anesthetized animal in which select nerve cells had been removed. He had trouble keeping the drugged salamanders alive, so maybe the anesthetic affected the outcome. But even the ones that survived didn't regenerate at all. The growth factor was almost certainly not acetylcholine. Vital electricity. These, then, were the shoulders on which I stood in 1958 as I began to look for the pattern control and the blastema stimulating factors in regeneration. At that time, we knew of two things that could yield some regrowth in non-regenerators, extra nerve and extra injury. How were they related? Luck gave me a clue. I began my work just after the first few Sputniks during the missile gap flap. Alarmed by unforeseen triumphs of Russian technology, which we consider primitive, the government hastily began translating every Soviet scientific journal and distributing copies free to federally funded research centers. Suddenly, the medical library at the VA Medical Center in Syracuse, where I worked, began receiving each month a crate of Russian journals on clinical medicine and biology. Since no one else was much interested, this bonanza was all for me. I soon made two discoveries. The Russians were willing to follow hunches. Their researchers got government money to try the most outlandish experiments, one that our scientists just knew couldn't work. Furthermore, Soviet journals published them, even if they did work. I particularly enjoyed Biophysica, Soviet Journal of Biophysics. And it was there I encountered a paper on the nature of the variation of the bioelectric potentials in the regeneration process of plants. By A.M. Sinyukin of Lomonosov State University in Moscow, Sinyukin began by cutting one branch from each of the series of tomato plants. Then he took electrical measurements around the wound as each plant healed and sent out a new shoot near the cut. He found a negative current, a stream of electrons flowing from the wound for the first few days. A similar current of injury is emitted from all wounds in animals. During the second week, after a callus had formed over the wound, the new branch had begun to form. The current became stronger and reversed its polarity to positive. The important point wasn't the polarity. The position of the measuring electrode with respect to the reference electrode often determines whether a current registers a positive or negative. Rather, Sinyukhin's work was significant because he found a change in the current that seemed related to reparative growth. Sinyukhin found a direct correlation between these orderly electrical events and biochemical changes. As the positive current increased, cells in the area more than doubled their metabolic rate, also becoming more acidic and producing more vitamin C than before. Sinyukhin then applied extra current using small batteries to a group of newly lopped plants. augmenting the regeneration current. These battery-assisted plants restored their branches up to 3 times faster than the control plants. The currents were very small, only 2 to 3 microamperes for 5 days. An ampere is a standard unit of electric current, and a microampere is 1/millionth of an ampere. Larger amounts of electricity killed the cells and no growth-enhancing effect. Moreover, the polarity had to match that normally found in the plant. When Senyunkin used current of the opposite polarity, nullifying the plant's own current, restitution was delayed by 2 or 3 weeks. To American biology, however, this was all nonsense. To understand why, we must backtrack for a while. Luigi Galvani, an anatomy professor in the medical school at the University of Bologna, who'd been studying electricity for 20 years, first discovered the current of injury in 1794, but unfortunately he didn't know it. At that time, biology's main concern was the debate between vitalism and mechanism. Vitalism, though not always called by that name, had been the predominant concept of life since prehistoric times throughout the world, and it formed the basis for almost all religions. It was closely related to Socrates and Plato's idea of supernatural forms, or ideals from which all tangible objects and creatures derived their individual characteristics. Hippocrates adapted this idea by postulating an anima as the essence of life. The Platonic concept evolved into the medieval philosophy of realism, whose basic tenet was that abstract universal principles were more real than sensory phenomena. Mechanism grew out of Aristotle's less speculative rationalism, which held that universal principles were not real, being merely the names given to humanity's attempts at making sense of the reality apprehended through the senses. Mechanism had become the foundation of science through the writings of Descartes in a previous century, although even he believed an animating force to give the machine life at the outset. By Galvani's time, mechanism influence was steadily growing. Galvani was a dedicated physician in medicine, tracing its lineage back to tribal shamans. Has always been a blend of intuition and empirical observation based on a vitalistic concept of sanctity of life. The vitalist had long tried unsuccessfully to link the strange, incorporeal phenomenon of electricity with the élan vital. This was Galvani's main preoccupation. One day he noticed that some frogs' legs he'd hung in a row on his balustrade pending his dinner twitched whenever he blew them against the ironwork. At about the same time, his wife Lucia noticed in his laboratory that the muscles of the frog's leg contracted when an assistant happened to be touching the main nerve with the steep scalpel at the same instant that a spark leaped from one of the electrical machines being operated across the room. The only type of electricity then known was the static type, in the form of sparks from various friction devices. Today, we know that an expanding and collapsing electric field generated by the spark induced a momentary current in the scalpel, which stimulated the muscle. But Galvani believed that the metal railing and scalpel had drawn forth electricity hidden in the nerves. Galvani experimented for years and with nerves from frogs' legs connected in various circuits with several kinds of metals. He grew convinced that the vital spirit was electricity flowing from the nerves and announced this to the Bologna Academy of Science in 1791. Within 2 years, Alessandro Volta, physicist of the University of Padua, had proven that Galvani had in fact discovered a new kind of electricity, steady current rather than sparks. He generated a biometallic direct current, a flow of electrons between two metals, such as the copper hooks and the iron railing of the famous balcony observation, connected by a conducting medium. In other words, a battery. frog's legs being more or less bags of weak salt solution where the electrolyte or conducting medium. They were otherwise incidental, Volta explained, and there was no such thing as Galvani's animal electricity. Galvani, a shy, thoroughly non-combative soul, was crushed. His only response was an anonymous paper in 1794 describing several experiments in which frog legs could be made to twitch with no metal in the circuit. In one procedure, the experimenter touched one leg nerve with the frog's dissected out naked spinal cord while holding the other leg to complete the circuit. Here, the current was true animal electricity coming from the amputation wound at the base of the leg. In the long run, Galvani unwittingly helped the cause of the mechanist by giving them something to attack. As long as the élan vital was ephemeral, all you could say was that you couldn't find it. Once Galvani said it was electricity, a detectable, measurable entity. There was a target for experimentation. Actually, Baron Alexander von Humboldt, the explorer naturalist who found geology, proved in 1797 that Volta and Galvani were both partly right. Biometallic currents were real, but so was spontaneous electricity from injured flesh. However, the mechanist had the upper hand. Galvani's anonymous report and Humboldt's confirmation were overlooked. Galvani himself died penniless and broken-hearted in 1798, soon after his home and property were confiscated by the invading French, while Volta grew famous developing his storage batteries under the auspices of Napoleon. Then in the 1830s, a professor of physics at Pisa, Carlo Matteucci, using the newly invented galvanometer, which could measure fairly small direct currents, came up with other evidence for animal electricity. In a meticulous series of experiments lasting 35 years, he conclusively proved the current of injury was real. However, he didn't find it in the nervous system, only emanating from the wound surface, so it couldn't be firmly related to the vital force. The tale took another turn in the 1840s when Emile Dubois Raymond, a physiology student in Berlin, read Matteucci's work. Dubois Raymond went on to show that when a nerve was stimulated, an impulse traveled along it. He measured the impulse electrically and announced his conclusion that it was a mass of electromotive particles, like a current in a wire. Immediately, he squared his shoulders, expecting the mantle of glory to descend. If I do not greatly deceive myself, he wrote, I have succeeded in realizing in full actuality the 100 years dream of physicists and physiologists, to wit, the identity of the nervous principle with electricity. But he had deceived himself. Soon it was learned that the impulse traveled too slowly to be a current, and that nerves didn't have the proper insulation or resistance to conduct one anyway. Any true current the size of a small measured impulse wouldn't have made it through even a short nerve. Julius Bernstein, a brilliant student of Dubois Raymond, resolved the impasse in 1868, which his hypothesis of the action potential, the impulse, wasn't a current, Bernstein said. It was a disturbance in the ionic properties of the membrane, and it was this perturbation that traveled along the nerve fiber or axon. The Bernstein hypothesis stated that the membrane could selectively filter ions of different charges to the inside or outside of the cells. Ions are charged particles into which a salt breaks up when dissolved in water. All salts dissociate in water into positive and negative ions, such as the positive sodium and negative chloride ions of table salt. Bernstein postulated that the membrane could sort most of the negatives outside and most of the positives inside the fiber. The membrane was polarized with like charges grouped on one side, having a transmembrane potential because the negative charges all on one side could potentially flow in a current across the membrane to achieve a balance on both sides. This was what happened in a short segment of the membrane whenever a nerve was stimulated. Part of the membrane became depolarized, reversing the membrane potential. The nerve impulse was actually a disturbance in the potential traveling along the membrane. As the area of disturbance moved along, the membrane quickly restored its normal resting potential. Thus, the nerve impulse wasn't an electrical current, even though it could be measured electrically. It is an area of reversed polarization in the nerve membrane that moves down the nerve fiber. It can be measured electrically, but there is no current flowing down the fiber. The nerve impulse. Bernstein's hypothesis had been confirmed in all important respects, although it remains a hypothesis because no one has yet found what gives the membrane the energy to pump all those ions back and forth. Soon it was broadened, however, to include an explanation of the current of injury, reasoning that all cells had been transmembrane potentials. Bernstein maintained that after injury, the damaged cell membranes simply leaked their ions into the environment. Thus, the current of injury was no longer a sign that electricity was essential to life, but only an uninteresting side effect of cell damage. The vitalists, with their hopes pinned on electricity, kept getting pushed into tighter and tighter corners as the electricity was removed from one part of the body after another. Their last stand occurred with the discovery of neurotransmitters. They'd maintained that only an electrical current could jump across the synapses, the gap between communicating nerves. In 1920, that idea was disproven with a lovely experiment by Otto Loewy, a research professor at NYU School of Medicine, later my alma mater. When I took physiology in my first year there, we had to duplicate his experiment. Stimulation of depressor nerve slows the heartbeat of a frog heart in a solution. A new heart is suspended in the solution used above and it beats slows automatically. Loewy's experiment, the discovery of acetylcholine. Biologists had found that a frog heart would continually beat for several days when removed with its nerves and placed in an appropriate solution. Stimulating one of the nerves would slow it down. Like Loewy, we took one such heart with nerve attached and stimulated the nerve, slowing the beat. We then collected the solution bathing the heart and placed another heart in it. Its beat slowed even though its depressor nerve hadn't been stimulated. Obviously, the nerve slowed the heartbeat by producing a chemical which crossed the gap between the nerve ending and the muscle fiber. This chemical was later identified as acetylcholine, and Loewy was awarded the Nobel Prize in 1936 for his discovery. His work resulted in the collapse of the last vestige of electrical vitalism. Thereafter, every function of the nervous system had to be explained on the basis of the Bernstein hypothesis and chemical transmission across the synapse. It was with great trepidation, therefore, that I put any credence in Sinyukin's report that the strength of injury current affected regeneration in his plants. Yet his report was detailed and carefully written. Something about his work gave me a gut feeling that it was valid, Maybe it was because the tomato plants he used were, best of all, American beauties. At this point, I wasn't aware of Matucci's forgotten work, but something clicked in my mind now that I studied Rosie's and Polichev's experiments, and both definitely in Polichev's and probably in Rose's regeneration had been stimulated by an increase in the injury. And another Russian supplied a timely lead. In a government translation, I found a 1958 paper by A.V. Zerbinsky of the Institute of Cytology in Leningrad, who studied the current of injury in the hind leg muscle of the bullfrog. This muscle is nice and long, easy to work with, and contains branches from several different nerves. He made a standard injury in each muscle, measured the current of injury, and then cut the nerves branch by branch, noting the effect on the current. It decreased with each succeeding nerve cut. The current of injury was proportional to the amount of nerve. Then I went to the library and delved back into the history of neurophysiology and found Matucci's superb series of observations. Not only one had he proven that the current of injury was real, he showed that it varied in proportion to the severity of the wound. Now I had enough pieces to start on the puzzle. I summarized the observations in a little matrix. Extent of the injury is proportional to the regeneration. Amount of the nerve is proportional to the regeneration. Extent of injury is proportional to current of injury. Amount of nerve is proportional to current of injury. Ergo, current of injury is proportional to regeneration. I was pretty sure now that contemporary knowledge to the contrary, the current of injury was no side effect and was the first place to look for clues to the growth control and dedifferentiation stimulating factors. I planned my first experiment. Chapter 3, The Sign of the Miracle. Real science is creative, as much as so as painting, sculpture, or writing. Beauty, variously defined, is the criterion for art. And likewise, a good theory has the elegance, proportion, and simplicity that we find beautiful. Just as the skilled artist omits the extraneous and directs our attention to a unifying concept, so the scientist strives to find a relatively simple order underlying the apparent chaos of perception. Perhaps because it was mine, my theory that the current of injury stimulated regeneration seemed both simple and beautiful. It's impossible to convey the sense of excitement I felt when all the facts fell together and the idea came. I'd created something new that explained the previously inexplicable. I couldn't wait to see if I was right. And all the time that the Bernstein hypothesis had been used to explain away the current of injury, no one had ever thought to measure the current over a period of days to see how long it lasted. If it was only ions leaking from damaged cells, it should disappear in a day or two, when these cells had finished dying or repairing themselves. The simple measurement with the compensation of the currents in regenerating versus non-regenerating limbs was what I planned to do. I would uniformly amputate the forelegs of the frogs and salamanders. Then, as the frog's stumps healed over and the salamander's legs regrew, I would measure currents of injury each day. The experiment itself was as simple as could be. The tricky part was getting permission to do it. When you want to do a research project, there are certain channels you must go through to get the money. You write a project proposal spelling out what hypothesis you want to test, why you think it should be done, and how you plan to go about it. The proposal goes to a committee supposedly composed of your peers, people who have demonstrated competence in related research. If they approve your project and the money is available, you generally get part of what you asked for, enough to get started. The Veterans Administration had been dispensing research money for years as a sort of bribe to attract doctors despite the low pay and government service. The money from Washington was doled out by the most influential doctors on the staff, not necessarily the best researchers. But I still felt I had a good chance because the VA was having an especially hard time recruiting orthopedists. Moreover, my hypothesis was based on the work of Rose, Pilichev, Singer, Sinyukhin, and Zermunsky with inescapable logic. And since frogs and salamanders were anatomically similar, any difference in their currents of injury should reflect a disparity in their powers of regeneration. My chances of being thrown off by extraneous factors were thus minimal. I remember thinking as I wrote the proposal how my life had come full circle. As a college freshman in 1941, I had conducted a crude experiment on salamanders, showing that thyroid stimulation by iodine didn't speed up regeneration. Here, I was nearly 20 years later, a beneficiary of the intervening research, hoping to add to our knowledge of the same phenomenon and perhaps even discover something that would help human patients. I worried that my roundabout course might weigh against me, since one of the criteria for grants was whether the investigator had been trained for that particular field. This proposal would have been expected from a physiologist, not an orthopedist. Nevertheless, I was asking for a relatively minuscule amount of money. I needed only $1,000 to put together the equipment, so I didn't anticipate much trouble. The Tribunal. Dr. Becker, could you please come to a special research committee meeting in 1 hour? Committee secretary was calling. I'd known something was up. 2 months had passed since I'd filed my proposal, and all my queries as to its fate had gone unanswered. I'll be there. It's not here in the research office. It's downstairs in the hospital director's office. Now that was really strange. The director almost never paid any attention to the research program. Besides, his office was big enough to hold a barbecue in. It was a barbecue, all right. And I was the one being grilled. The director's conference room had been rearranged. In place of the long polished table, there was a semicircle about a dozen chairs, each occupied by one of the luminaries from the hospital and medical school. I recognized the chairman of the departments of biochemistry and physiology, along with the hospital director and the chief of research. Only the dean was missing. In the center was a single chair for me. The spokesman came right to the point. We have a very grave basic concern over your proposal. This notion that electricity has anything to do with the living was totally discredited some time ago. It has absolutely no validity, and the new scientific evidence you're citing is worthless. The whole idea was based on its appeal to quacks and the gullible public. I will not stand idly by and see this medical school associated with such a charlatanistic, unscientific project. Murmurs of assent spread through the group. I had the momentary thrill of imagining myself as Galileo or Giordano Bruno. I thought of walking to the window to see if the stake and the faggots were set up on the lawn. Instead, I delivered a terse speech to the effect that I still thought my hypothesis was stoutly supported by some very good research, and that I was sorry if it flew in the face of dogma. I ended by saying that I didn't intend to withdraw the proposal, so they would have to act upon it. When I got home, my fury was gone. I was ready to call the director, withdraw my proposal, apologize for my error, stay out of research, quit the VA, and go into private practice where I could make a lot more money. Luckily, my wife Lil knows me better than sometimes I know myself. She told me, "You'd be miserable in private practice. This is exactly what you want to do. So just wait and see what happens." 2 days later, I got the word that the committee had delegated this decision to Professor Chester Yintema, an anatomist who long ago had studied the regrowth of ears in the salamander. Since he was the only one in Syracuse who'd ever done any regeneration research, I've always wondered why he wasn't part of the first evaluation. I went to see him with a sense of foreboding. for his latest research seemed to refute Singer's nerve work, on which I'd based my proposal. Using a standard technique, Yuntema had operated on very young salamander embryos, cutting all of the tissues that would have given rise to the nervous system. He then grafted each of these denervate embryos onto the back of a whole one. The intact embryos furnished the grafts with blood and nourishment, and the procedure resulted in a little parabiotic twin, normal except for having no nerves, stuck on the back of each host animal. Yuntema then cut off one leg from each of these twins, and some of them regenerated. Since microscopic examination revealed no nerves entering the graft from its host, Yuntema's experiment called Singer's conclusions into question. Dr. Yuntema turned out to be one of the nicest gentlemen I've ever met, but as I entered his office in Dickinson appearance of eminence, he was tall, thin, elderly, with craggy features, and wore an immaculately starched, long white coat lab. Made me feel like a freshman being called before the dean. But he put me at ease immediately. "I've read your proposal and think it's most intriguing," he said with genuine interest. "Do you really?" I asked. "I've been afraid you would reject it out of hand because my ideas depend on Singer's work." "Mark Singer is good, careful," Yuntema replied. "I don't doubt his observations. What I've described is an exception to his findings under special circumstances." After a long, pleasant conversation about regrowth, nerves, and research itself, he gave me his approval with a word of caution. "Don't get your hopes up about what you want to do. I don't believe for 1 minute that it'll work, but I think you should do it anyway. We need to encourage young researchers. Besides, it'll be fun, and maybe you'll learn something new after all. Let me know what happens, and if you need any help, I'll be there. I'll call the people at the VA right away, so get to work. Good luck." This was the start of a long friendship. I'm deeply indebted to Chester Yuntema for his encouragement. Had he not believed that research should be fun, that you should do what you want rather than what's fashionable, my first experiments would have been impossible, and this book would have never been written. The reversals. First, I found a good supplier of salamanders and frogs, a Tennessee game warden who ran this business in his spare time. Sometimes the shipment would contain a surprise, a small snake. I never found out whether he included them deliberately or by error. At any rate, his animals weren't the inferior aquarium bred stock, but robust specimens collected from their natural habitats. Next, I worked out some technical problems. The most important of these was the question of where to place the electrodes. To form the circuit, 2 electrodes had to touch the animal. One was the hot or measurable electrode, which determined the polarity, positive or negative. With regard to stationary reference electrode, a negative polarity meant that there were more electrons where the measuring electrode was placed, while positive polarity meant there were more at the reference site. A steady preponderance of negative charge at a particular location could mean there was a current flowing toward that spot, continually replenishing accumulation of electrons. The placement of reference electrode, therefore, was critical, lest I get the voltage right but polarity, and hence the direction of the current wrong. Some logical position had to be chosen and used every time, since I postulated that the nerves were somehow related to the current. The cell bodies that sent their nerves fibers into the limbs seemed like a good reference point. These cell bodies were in the section of the spinal cord called the brachial enlargement, located just headward from where the arm joined the body. In both frogs and salamanders, therefore, I put measuring electrode directly on the current surface of the amputation stump and the reference electrode on the skin over the brachial enlargement. After setting up the equipment, I did some preliminary measurements on the intact animals. They all had areas of positive charge at the brachial enlargement and negative charge of about 8 to 10 millivolts at each extremity, suggesting a flow of electrons from the head and trunk out into the limbs and in the salamander's tail. I began the actual experiment by amputating the right forelimbs between elbow and wrist from 14 salamanders and 14 grass frogs, all under anesthesia. I took no special precautions against bleeding since blood clots formed rapidly. The wounds had to be left open, not only because the closing skin over the salamander's amputation sites would have stopped regeneration, but also because I was investigating a natural process. In the wild, both frogs and salamanders get injuries much like the ones I was producing. Both are favorite foods of the freshwater bass and heal them without a surgeon. Once the anesthetic wore off and the blood clot formed, I took a voltage reading from each stump. I was surprised to find that the polarity at the stump reversed to the positive right after the injury, but by the next day it climbed to over 20 millivolts, the same in both frogs and salamanders. I made measurements daily, expecting to see the salamander voltages limb above those of the frog as the blastemas formed. It didn't work that way. The force of the current flowing from the salamander amputation sites rapidly dropped. While that formed, the frog stumps stayed at the original level. By the third day, the salamanders showed no current at all, and their blastemas hadn't even begun to appear. The experiment seemed a failure. I almost quit right there, but something made me keep on measuring. I guess I thought it would be good practice. Then, between the 6th and 10th days, an exciting trend emerged. The salamander potentials changed their sign again, exceeding their normal voltage and reaching a peak of more than 30 millivolts negative just when the blastemas were emerging. The frogs were still plugging away with slowly declining positive voltages. As the salamander limbs regenerated and the frog stumps healed over with skin and scar tissue, both groups of limbs gradually returned from opposite directions to the original baseline of 10 millivolts negative. The current of injury, salamander versus frog. Here was confirmation better than my wildest dreams. Already in my first experiment, I had the best payoff research can give. The excitement of seeing something no one else had ever seen before. I knew now that the current of injury wasn't due to dying cells, which were long gone by then. Moreover, the opposite polarities indicated a profound difference in the electrical properties of the two animals, which somehow would explain why only the salamander could regenerate. The negative potential seemed to bring forth the all-important blastema. It was a very significant observation, even though the facts had scrambled my near hypothesis somewhat. The current of injury is more than a side effect. Dr. Intima agreed and urged me to write up a report for publication. But first I jumped ahead with another idea. I took a new group of frogs, amputated one frog leg from each, and every day I applied negative current to the stump from a small battery. In the graphic, it says, simple negative charges from damaged cells. In the next graphic, it says, soon balanced by positive charges. And in the last graphic, it says, there must be a continuous current. I dreamed of being the first to get complete regrowth in normally non-regenerating animals. I could almost see my name on the cover of Scientific American. The frogs were less interested in my glory. They had to hold still for up to half an hour with electrodes attached. They refused, so I anesthetized them each day, something they tolerated very poorly. Within a week, my Nobel Prize had turned into a collection of dead frogs. For some time, I had been scouring the dusty stacks of the medical library for previous work on bioelectricity, and now I found a paper written in 1909 by an American researcher named Owen E. Frazee, who reported that electrical currents passed through the aquarium water in which larval salamanders were living, speeding up their regeneration. At that time, electrical equipment was so primitive that I couldn't rely on Frazee's results, but I decided to try it for myself. What Senyakin had done with tomato plants, I hoped to do with salamanders. To one group of salamanders, I applied 2 microamperes of positive current from batteries connected directly to the stumps for 5 to 10 minutes on each of the first 5 days after amputation. This was 0.0000002 ampere, a tiny current by ordinary standards. Most household circuits carry 15 or 20 amperes. But comparatively to what seemed to be flowing in the limb, I intended to reinforce normal positive peak in the current of injury. This treatment seemed to make the blastemas larger but slowed down the whole process somewhere. To another group, I applied 3 microamperes of negative current on the 5th to the 9th days when the normal currents were hitting their negative peaks. This seemed to increase the rate of regrowth for a week, but didn't change the time needed for a complete limb. Finally, I tried Frazee's method with a constant current through the aquarium water. Again, the results were equivocal at best. These failures taught me that before I applied my findings to other animals, I would have to learn how the current of injury worked. Meanwhile, I wrote up my results. Not knowing any better, I submitted my paper to the Journal of Bone and Joint Surgery, the most prestigious orthopedic journal in the world. It was a dumb thing to do. The experiment had no immediate practical application, while the journal accepted only clinical reports. Moreover, the publication was very political. Normally, you had to have an established reputation or come from one of the big orthopedic programs like Harvard or Columbia to get into it. Luckily, I didn't know that. Someone thought my paper was just what the doctor ordered. Not only was it accepted for publication, but I was invited to present it at the next combined meeting of the Orthopedic Research Society and the American Academy of Orthopedic Surgeons at Miami Beach in January 1961. This invitation was a particular honor, for admit someone considered my work so significant that practicing physicians as well as researchers should hear of it right then and there. Whoever that someone was, he or she has my underlying gratitude. My report was well received and soon was published. To the consternation of the local inquisitors and the delight of Chester and Intema. Since the journal was geared to clinicians, I worried that my experiment wouldn't reach the basic researchers with whom I really wanted to share it. But again, I was wrong. The next year, I got a phone call from Merrill Rose himself. He was excited by the article and wanted to know what I'd done since. Although Rose taught at Tulane Medical School in New Orleans, he spent every summer at the Woods Hole Marine Biological Library on Cape Cod, so he and his wife drove to Syracuse from there. Despite his success, Rose had maintained the completely open mind that a great researcher must have, and he was fascinated by the observations on electric fields, nerves, anesthesia, and magnetism that I'll recount in my next chapter. Since then, his interest has encouraged me enormously. My friendship with his fine man and scientist has been fruitful, even beyond the expectations I had then. And when my wife and I had the Roses to dinner, we found our paths were linked by an odd coincidence. As they walked in the door, Lillian exclaimed, Dr. Rose, weren't you at Smith's College in 1940s? It turned out that she'd been a friend of Rose's student lab assistant and had helped catch the frogs for the famous salt in the wound experiment. Part 2. The stimulating current. The basic texture research consists of dreams into which the threads of reasoning, measurement, and calculation are woven. Quote by Albert Szent-Györgyi. Chapter 4. Life's Potentials. It's an axiom of science that the better an experiment is, the more new questions it raises after its answer, the one you need asked. By that standard, my first simple test had been pretty good. The new problems branched out like the fingers on those restored limbs. Where did injury currents come from? Were they, in fact, related to the nervous system? And if so, how? It seemed unlikely that they sprang into action only after the amputation. They must have existed before. There must have been a pre-existing substratum of direct current activity that responded to the injury. Did the voltages I measured really reflect such currents? And did they flow throughout the salamander's body? Did other organisms have them? What structures carried them? What were their electrical properties? What were they doing the rest of the time, before injury and after healing? Could they be used to provoke regeneration where it was normally absent? I had ideas about how to look for some of the answers, but to understand my approach, the reader unfamiliar with electrical terms will need a simplified explanation of several basic concepts that are essential to the rest of the story. Everything electrical seems from the phenomenon of charge. No one knows exactly what this is, except to say that it's a fundamental property of the matter that exists in two opposite forms, or polarities, which we arbitrarily call positive and negative. Protons, which are one of the two main types of particles in atomic nuclei, are positive, and the other particles, the neutrons, are so named because they have no charge. Orbiting around the nucleus are the electrons, in the same number as the protons inside the nucleus. Although an electron is 1,836 times less massive than a proton, the electron carries an equal but opposite, or negative, charge. Because of their lightness and their position outside the nucleus, electrons are much more easily dislodged from atoms than are protons, so they're the main carriers of electric charge. For the layperson's purpose, a negative charge can be thought of as a surplus of electrons, while a positive charge can be considered a scarcity of them. When electrons move away from an area, it becomes positively charged, and the area to which they move becomes negative. The flow of electrons is called a current, and is measured in amperes, units named for an early 19th century French physicist, André Marie Ampere. A direct current is more or less even flow, as opposed to the instantaneous discharge of static electricity as sparks or lightning, and the back and forth flow of alternating current, which powers most of our appliances. Besides the amount of charge being moved, the current has another characteristic important for our narrative: its electromotive force. This can be visualized as the push behind the current, and it's measured in volts, named for Alessandro Volta. In high school, most of us learned that the current flows only when a source of electrons, or negatively charged material, is connected to a material having fewer free electrons, positively charged in relation to the source, by a conductor, through which the electrons can flow. This is what happens when you connect the negative terminal of a battery to its positive pole with the wire or a radio's innards. You've completed a circuit between negative and positive. If there's no conductor and hence no circuit, there's only hypothetical charge flow or electric potential between the two areas. The force of this latent current is also measured in volts by temporarily completing the circuit with a recording device, as I did in my experiment. The potential can continue to build until a violent burst of current equalizes the charges. This is what happens when lightning strikes. Smaller potentials remain stable, however. In this case, they must be continuously fed by a direct current flowing from positive to negative, the opposite of the normal direction. In this part of the circuit, electrons actually flow from whether they're scarce to where they're more abundant. As Volta found, such a flow is generated inside a battery by the electrical interaction of two metals. An electric field forms around any electric charge. This means that any charge object will be attracted if the polarities are opposite, or repelled if they're the same, for a certain distance around the first object. The field is the region of space in which an electrical charge can be detected, and it's measured in volts per unit of area. Electric fields must be distinguished from magnetic fields, like charge. Magnetism is a dimly understood intrinsic property of matter that manifests itself in two polarities. Any flow of electrons sets up a combined electric and magnetic field around the current, which in turn affects other electrons nearby. Around a direct current, the electromagnetic field is stable, whereas an alternating current's field collapses and reappears with its poles reversed every time the current changes direction. This reversal happens 60 times a second in our normal house currents. Just as current produces a magnetic field, when it moves in relation to a conductor, induces a current. Any varying magnetic field, like that around household appliances, generates a current in nearby conductors. The weak magnetic fields we'll be discussing are measured in Gauss, units named after a 19th century German pioneer in the study of magnetism, Carl Friedrich Gauss. Both electric and magnetic fields are really just abstractions that scientists have made up to try to understand electricity's and magnetism's action at a distance, produced by no known intervening material or energy, a phenomenon that used to be considered impossible until it became undeniable. A field is represented by lines of force, another abstraction to indicate its direction and shape. Both kinds of fields decline with distance, but their influence is technically infinite. Every time you use your toaster, the fields around it perturb charged particles in the farthest galaxies ever so slightly. In addition, there's a whole universe full of electromagnetic energy, radiation that somehow seems to be both waves in an electromagnetic field and particles at the same time. It exists in a spectrum of wavelengths that includes cosmic rays, gamma rays, X-rays, ultraviolet radiation, visible light, infrared radiation, microwaves, and radio waves. Together, electromagnetic fields and energies interact in many complex ways that have given rise to much of the natural world, not to mention the whole technology of electronics. You'll need a casual acquaintance with all these terms for the story ahead, but don't worry if the concepts seem a bit murky. Physicists have been trying for generations to solve the fundamental mysteries of electromagnetism, and no one, not even Einstein, has yet succeeded. They measured changes in these potentials and correlated them to growth, regeneration, tumor formation, drug effects, hypnosis, and sleep. Burr claimed to have measured field changes resulting from ovulation, but others got contradictory results. He hooked up his volunteers to trees for years at a time, found that their fields varied in response not only to light and moisture, but to storms, sunspots, and the phases of moon as well. Burr and Lund were handicapped by their instruments as well. As the research climate, most of their work was done before World War II, and even though Burr spent years designing the most sensitive devices possible using vacuum tubes, the meters were still too noisy to reliably measure the tiny currents found in living things. The two scientists could refine their observations only enough to find simple dipole distribution of potentials, the head of most animals being negative and the tail positive. Burr and Lund advanced similar theories of an electrodynamic field called by Burr the field of life, or the L field, which held the shape of an organism just as the mold determines the shape of a gelatin dessert. When we meet a friend we have not seen for 6 months, there is not one molecule in his face which was there when we last saw him, Burr wrote. But thanks to his controlling L field, the new molecules have fallen into the old, familiar pattern, and we can recognize his face. Burr believed that faults in the field could reveal latent illness, just as dents in a mold show up in a jelly. He claimed to be able to predict all sorts of things about a person's emotional and physical health, both present and future, merely by checking the voltage between head and hand. His later writings were marred by a sort of bioelectric determinism and tendency to confuse law and order in nature with that odious euphemism as preached by presidents. As a result, he began to suggest his simple readings as a foolproof way to evaluate job applicants, soldiers, mental patients, and suspected criminals or dissidents. The fields Burr and Lund found were actually far too simple to account for a salamander's limb or a human face. Biological knowledge at that time gave them no theoretical framework to explain where their fields came from. They conceived of currents flowing within cells, but had no proof. They had no inkling that currents might flow in specific tissues or in the fluids outside cells. They suggest that all these little intracellular currents somehow added up to the whole field. Burr wrote that electrical energy is fundamental attribute of protoplasm and is an expression or measure of the presence of electrodynamic field in organism. Unfortunately, analysis of this sentence yields nonsense, and Burr's work was dismissed as a foggy vitalism. Lund suffered the same fate. No one bothered to see if the measurements they had made were valid. After all, you can disagree with the theory, but you should respect the data enough to check them. If you can't duplicate them, you're entitled to rest easy with your own concepts. But if you get the same results, you're obligated to agree or propose an alternate theory. Most scientists took the easy way out, however, and simply ignored Burr and Lund. Their discoveries remained little known, and most biologists didn't connect them with the tentative morphogenic field concept of regeneration. Then in 1952, Lund's work was taken up by G. Marsh and H.W. Beams using the planarian. They found that the flatworm's polarity, like the hydra's, could be controlled by passing a current through it. When a direct current was fed in the proper direction through a section of a worm, normal polarity disappeared and head formed at each end. As the current strength was increased, the section's polarity reversed. A head regrew at the rear. a tail at the front. At higher voltages, even intact worms completely reorganized, with the head becoming a tail and vice versa. Marsh and Beams grew convinced that the animal's electric field was morphogenic organizing principle. Still, their work was also ignored, except by Merrill Rose, who suggested that a gradation of electrical charge from front to back controlled the gradient of growth inhibitors and stimulators. He suggested that the growth compounds were charged molecules that were moved to different places in the body by the electric field, depending on the amount and sign of their charge and their molecule weight. While the investigation of the total body field moved haltingly forward in the study of simple animals, several neurophysiologists began finding out odd things about the nerves of more complex creatures, data that Bernstein's action potential couldn't explain. Going through the old literature, following lead after lead from one paper to the next, I found many hints that there were DC potentials in the nervous system and that small currents from outside could affect brain function. The first recorded use of currents on the nervous system was by Giovanni Aldini, a nephew of Galvani, and an ardent champion of vitalism. Using the batteries of his archenemy, Volta, Aldini claimed remarkable success in relieving asthma. He also cured a man who today would probably be diagnosed as schizophrenic, although it's impossible to know how much benefit came from the currents and how much from simple solicitude, then so rare in treating mental illness. Aldini gave his patient a room in his own house and later found him a job. Some of Aldini's experiments were grotesque. He tried to resurrect recently executed criminals by making the corpse twitch with electricity. But his idea that external current could replenish the vital force of exhausted nerves became the rationale for a whole century of electrotherapy. Modern studies of nerves and current began in 1902 when French researcher Stéphane Leduc reported putting animals to sleep by passing fairly strong alternating currents through their heads. He even knocked himself unconscious several times by this method. Talk about dedication to science. Several others took up this lead in the 1930s and developed the technique of electroshock and electronarcosis. The therapeutic value of using large currents to produce convulsions had been questioned more and more until now. It's mostly used to quiet unmanageable psychotics and political nonconformists. Electronarcosis, induction of sleep by passing small currents across the head from temple to temple, is widely used by legitimate therapists in France and the Soviet Union. Russian doctors claim their electroson technique, which uses electrodes on the eyelids and behind the ears to deliver weak direct currents pulsing at commutative brainwave frequencies, can impart the benefits of a full night's sleep in 2 or 3 hours. There's still much dispute about how both techniques work, but from the outset, there was no denying that the currents had a profound effect on the nervous system. In the second and third decades of this century, there was a flurry of interest in galvanotaxis, the idea that direct currents guided the growth of cells, especially neurons. In 1920, S. Ingvar found that the fibers growing out of nerve cell bodies would align themselves with a nearby flow of current, that the fibers growing toward the negative electrode were different from those growing toward the positive one. Paul Weiss soon explained this heretical observation as an artifact caused by stretching of the cell culture substrate due to contact with the electrodes. Even after Marsh and Beams proved Weiss was wrong in 1946, It took many more years for the scientific community to accept the fact that neuron fibers do orient themselves along a current flow. Today, the possible use of electricity to guide nerve growth is one of the most exciting prospects in regeneration research. See Chapter 11. The Bernstein hypothesis, unable to account for these facts, has turned out to be deficient in several respects. To begin with, according to the theory, an impulse should travel with equal ease in either direction along the nerve fiber. If the nerve is stimulated in the middle, an impulse should travel in both directions to opposite ends. Instead, impulses travel only in one direction in experiments that can be made to travel upstream, but only with great difficulty. This may not seem like such a big deal, but it is very significant. Something seems to polarize the nerve. Another problem is the fact that although nerves are essential for regeneration, the action potentials are silent during the process. No impulses have ever been found to be related to regrowth. The neurotransmitters such as acetylcholine have been ruled out as growth stimulators. In addition, impulses always have the same magnitude and speed. This may not seem like such a big thing either, but think about it. It means the nerve can only carry one message, like the digital computers 1 or 0. This is okay for simple things like the knee-jerk reflex. When the doctor's rubber hammer taps your knee, it's actually striking the patellar tendon, giving it a quick stretch. This stimulates stretch receptors, nerve cells in the tendon, which fire a signal to the spinal cord saying, the patellar tendon has suddenly been stretched. These impulses are received by motor muscle activating neurons in the spinal cord, which sends impulses to the large muscle on the front of the thigh, telling it to contract and straighten the leg. In everyday life, the reflex keeps you from falling in a heap if an outside force suddenly bends your knees. The digital impulse system accounts for this perfectly well. However, no one can walk on reflexes alone. As victims of cerebral palsy know all too well, the motor activities we take for granted, getting out of a chair and walking across the room, picking up a cup and drinking coffee, and so on, require integration of all the muscles and sensory organs working smoothly together to produce coordinated movements that we don't even have to think about. No one has ever explained how the simple code of impulses can do all that. Even more troublesome are the higher processes such as sight, in which somehow we interpret a constantly changing scene made of innumerable bits of visual data, or the speech patterns, symbol recognition, and grammar of our languages. Heading the list of riddles is the mind-brain problem of consciousness with its recognition. I am real. I think. I am something special. Then there are abstract thought, memory, personality, creativity, and dreams. The story goes on that Otto Lowy had wrestled with the problem of synapses for a long time without result, when one night he had a dream in which the entire frog heart experiment was revealed to him. When he awoke, he knew that he had had the dream, but he'd forgotten the details. The next night he had the same dream. This time he remembered the procedure, went to his lab in the morning, did the experiment, and solved the problem. Inspiration that seemed to banish neural activity forever, neural electricity forever, can't be explained by theory it's supported. How do you convert simple digital messages into these complex phenomena? Latter-day mechanists have simply postulated brain circuitry so intricate that we will probably never figure it out, but some scientists have said there must be other factors. Even as Lowy was finishing his work on acetylcholine, others began to find evidence that currents flowed in the nerves. English physiologist Richard Canton had already claimed he directed an electric field around the heads of animals in 1875. But it wasn't until 1924 that German psychiatrist Hans Berger proved it by reporting the first electroencephalogram, or EEG, from platinum wires he inserted into a sun scalp. The EEG provided a recording of arithmetic function fluctuations in potential voltage over various parts of the head. Berger at first thought there was only one wave from the whole brain, but it soon became clear that the waves differed depending on where the electrodes were put. Modern EEG uses as many as 32 separate channels all over the head. The frequency of these brain waves has been crudely correlated with the states of consciousness. Delta waves are 0.5 to 3 cycles per second, indicate deep sleep. Theta waves, 4 to 8 cycles per second, indicate trance, drowsiness, or light sleep. Alpha waves, 8 to 14 cycles per second, appear during relaxed wakeful meditation, and beta waves, 14 to 35 cycles per second. The most uneven forms accompany all the modulations of our active everyday conscious. Underlying these rhythms are potentials that vary much more slowly. Over periods as long as several minutes, today's EEG machines are designed to filter them out because they cause the trace to wander and are considered insignificant anyway. There's still no consensus as to where EEG voltages come from. They would be mostly easily explained by direct currents, both steadily state and pulsing through the brain, but that has been impossible for most biologists to accept. The main alternative theory that large numbers of neurons firing simultaneously can mimic real electrical activity has never been proven. In 1939, W.E. Burge of the University of Illinois found that the voltage measured between the head and other parts of the body became more negative during physical activity, declined in sleep, and reversed to positive under general anesthesia. At about the same time, a group of physiologists and neurologists at Harvard Medical School began studying the brain with a group of MIT mathematicians. This association was destined to change the world. From it came many of our modern concepts of cybernetics, and it became the nucleus of the main American task force on computers in World War II. One of the group's first important ideas was that the brain worked by a combination of analog and digital coding. One of the mathematicians, computer pioneer John von Neumann, later elaborated on the concept in great detail. But basically, it's rather simple. In analog computers, changes in information are expressed by analogous changes in magnitude of polarity of current. For example, if a computer is to use and store the varying temperatures of a furnace, the rise and fall in heat can be mimicked by a rise and fall in voltage. Analog systems are slow and can handle only simple information, but they can express subtle variations as well. Digital coding, on the other hand, can transmit enormous amounts of data at high speed, but only if the information can be reduced to yes, no, on, off bits, the digits 1 and 0. If the brain was such a hybrid computer, these early cyberneticists reasoned, then analog coding could control the overall activity of large groups of neurons by such actions as increasing or decreasing their sensitivity to incoming messages. A few years later, neurologists did find that some neurons were turned into fire only if they received a certain number of impulses. The digital system would transfer sensory and motor information, but the processes of that information, memory and recall, thought, and so on, would be accomplished by the synergism of both methods. The voltage changes Burge found in response to major alterations of consciousness seemed to fit within this framework, and his observations were extended by the Harvard MIT group and others. Much of this work was done directly on the exposed brains of animals and human patients during surgery. When cooperation When cooperative, patients elected to remain awake during such operations. The brain is immune to pain. Human sensations could often be correlated with electrical data. Contributors to this endeavor include nearly all of the greatest American neurophysiologists: Walter Buchanan, Arturo Rosenbluth, Ralph Gerard, Gilbert Ling, Walter Penfield, and others. Measurements on this exposed brain quickly confirmed the existence of potential voltages and also revealed possible currents of injury. Whenever groups of nerve cells were actually actively conducting impulses, they also produced a negative potential. Positive potentials appeared from injured cells. When the brain was damaged, these potentials then expanded outward to uninjured cells, suppressing their ability to send or receive impulses. When experimenters applied small negative voltages to groups of neurons, their sensitivity increased. That is, they would generate an impulse in response to weaker stimulus. Externally applied positive potentials worked in the opposite way. They depressed nerve function, making it harder to produce an impulse. Thus, there did seem to be an analog code. But how did it work? Did the potentials come from direct currents generated by the nerve cells themselves? Or did they merely result from adding up a lot of action potentials, all going in the same direction and arriving in the same place at the same time? Some answers were provided by a series of beautiful experiments by Ling, Gerard, and Benjamin Libet at the University of Chicago. Working on frogs, they studied areas of the cortex where the neuron layer was only 1 cell thick and the cells were arranged by side like soldiers on review, all pointing in the same direction. In such areas, they found a negative potential on the dendrites, the short incoming fibers, and a positive potential at the ends of the axons, the longer outgoing fibers. This indicated a steady, direct current along the normal direction of impulse transmission. The entire nerve cell was electrically polarized. In another series of experiments on brains removed from frogs and kept alive in culture, the Chicago group found that the direct current swept across the surface of the cortex in very slow waves, which could be produced experimentally by applying chemicals such as caffeine to a single spot on the surface. When they made a cut on the brain, severing groups of nerve fibers, these DC traveling waves would still cross the cut if two surfaces were in direct contact. If the researchers held the cut open and filled it with a saline solution that matched body fluids, then the waves couldn't cross the gap. These were particularly important in observations. They indicated that the current was transmitted by structures outside the neurons. It crossed the cut when the edges touched, but the microscopic parts of several severed neurons wouldn't have rejoined so easily. The results showed that the current was not a flow of ions. Otherwise, it would have been able to cross the gap through the salt water. Studying intact brains in living frogs, the same group found a potential between the front and back of the brain. The olfactory frontal lobes were several millivolts negative with respect to the occipital rear lobe, implying the current flowing up the brain stem and between the two hemispheres to the front. At the time, these observations seemed mighty odd. They didn't fit any concepts of how the nerves worked. As a result, they were largely ignored. The majority of neurophysiologists weren't on measuring the action potentials and tracing out fiber pathways in the brain. This was useful, but worked limited. The basic questions remained. Only one research team followed up this work some 10 years later. Sidney Goldring and James L. O'Leary, neuropsychiatrists at the Washington University School of Medicine in St. Louis, recorded the same DC potentials from the human scalp. from the exposed brain during surgery and from the brains of monkeys and rabbits. As noted before, the potentials varied in regular cycles several minutes long, like a basso continuo under the EEG. In fact, Goldring and O'Leary found waves within waves, written upon the slow major swings or lesser voltage changes. These were weak potentials measured in microvolts, millionths of a volt, and varying in waves 2 to 30 cycles per minute of a pianissimo inner voice and three-part electrical fugue. Conducting in a new mode. I was actually aware that I didn't have the proper background for the work I planned to do. I wasn't a professional neurophysiologist. I didn't even know one. Indeed, after my run-in with the research committee, one member had taken me aside and earnestly advised me, go back to school and get your PhD, Becker. Then you'll learn all of this stuff is nonsense. Still, some of the greatest neurophysiologists had thought the same way I did about all of this stuff. They suggested we might have been too hasty in throwing electric currents of our biology. My notion of putting them back in wasn't so outlandish, but only an extension of what they'd been saying. I was approaching the body's system of information transfer from the periphery, asking, what makes wounds heal? They'd started from the center asking, how does the brain work? We were working on the same problem from opposite ends. As I contemplated their findings of all biology's unsolved problems, I grew convinced that life was more complex than we suspected. I felt that those reduced life into mechanical interaction of molecules were living in a cold, gray, dead world, which, despite its drabness, was a fantasy. I didn't think electricity would turn out to be any élan vital in the old sense, but I had a hunch it would be closer to the secret than the smells of the biochemistry lab or the dissecting room's preserved organs. I had another worthy ally when I started to reevaluate the role of electricity in life. Albert Szent-Györgyi, who'd already won a Nobel Prize for his work on oxidation in vitamin C, made a stunning suggestion in a speech before the Budapest Academy of Science on March 21st, 1941. Think of the date. World War II was literally exploding around him, and there he was, calmly laying the foundations for a new biology. Speaking of the mechanistic approach of biochemistry, he pointed out that when experimenters broke living things down into their constituent parts, somewhere along the line, life slipped through their fingers and they found themselves working with the dead matter. He said, it looks as though some if basic fact about life were still missing, without which any real understanding is impossible. For the missing basic fact, Szent-Györgyi proposed putting electricity back into the living things, but not the way it had been thought of at the turn of the century. At that earlier time, there had been only two known modes of current conduction, ionic and metallic. Metallic conduction can be visualized as a cloud of electrons moving along the surface of metal, usually a wire. It can be automatically excluded from living creatures because no one has ever found any wires in them. Ionic current is conducted in solutions by the movement of ions. Atoms are molecules charged by having more or fewer than the number of electrons needed to balance their protons, positive charges. Since ions are much bigger than electrons, they move more laboriously through the conducting medium, and ionic currents die out after short distances. They work fine across the thin membrane of the nerve fiber, but it would be impossible to sustain an ionic current down the length of the shortest nerve. Semiconduction, the third kind of current, was a laboratory curiosity in 1930s. Halfway between conductors and insulators, the semiconductors are inefficient in the sense that they can carry out only small currents, but they can conduct their currents readily over long distances. Without them, modern computers, satellites, and all the rest of our solid state electronics would be impossible. Semiconduction only occurs in materials having orderly molecular structure, such as crystals, in which electrons can move easily from the electron cloud around one atomic nucleus to the cloud around another. The atoms in a crystal are arranged in neat geometrical lattices rather than the frozen jumble of ordinary solids. Some crystalline materials have spaces in the lattice where other atoms can fit. The atoms of these impurities may have more or fewer electrons than the atoms of lattice material. Since the forces of lattice work structure hold the same number of electrons in place around each atom, the extra electrons of the impurity atoms are free to move through the lattice without being bound to any particular atom. If the impurity atoms have fewer electrons than the others, the holes in their electron clouds can be filled by electrons from other atoms, leaving holes elsewhere. A negative current or N-type semiconduction amounts to the movement of excess electrons. A positive current or P-type semiconduction is the movement of these holes, which can be thought of as positive charges. Three ways of conducting electrical current. Szent-Györgyi pointed out that molecular structure of many parts of the cell was regular enough to support semiconduction. This idea was almost completely ignored at the time. Even when Szent-Györgyi expanded the concept in his 1960 Introduction to Submolecular Biology, most scientists, except in Russia, dismissed it as evidence of advancing age, but that little book was inspiration to me. I think it may turn out to be the man's most important contribution to science. In it, he conjectured that protein molecules, each having a sort of slot or way station for mobile electrons, might be joined together in long chains so the electrons could flow in a semiconducting current over long distances without losing energy. Much as in a game of checkers, one counter could jump along a row of other pieces across the entire board. Suggested that the electron flow could be similar to photosynthesis, another process he helped elucidate, in which a kind of waterfall or electrons cascaded step by step down a staircase of molecules, losing energy with each bounce. The main difference was in protein semiconduction with the electron's energy would be conserved and passed along as information instead of being absorbed and stored in the chemical bonds of food, which sends Györgyi's suggestion in mind. I put together my working hypothesis. I postulated a primitive analog coded information system that was closely related to the nerves, but not necessarily located in the nerve fibers themselves. I theorized that this system used semiconductance directing currents and that either alone or in concert with the nerve impulse system, it regulated growth, healing, and perhaps other basic processes. Testing the concept. The first order of business was to repeat Burr's measurements on salamanders using modern equipment. I put the reference electrode at the tip of each animal's nose, moving the recorded electrode point along the center of the body of the tip of the tail, and then out along each limb. I measured voltages on the rest of the body and plotted lines of force connecting all points where the readings were the same. Instead of Burr's simple head negative and tail positive form, I found a complex field that followed the arrangement of the nervous system. There were large positive potentials over each lobe of the brain and slightly smaller ones over the branchial and lumbar nerve ganglia between each pair of limbs. The readings grew increasingly negative as I moved away from these collections of nerve cell bodies. The hands, feet, and tip of the tail had the highest negative potentials. In another series of measurements, I watched the potentials develop along with the nervous system in larval salamanders. In the adults, cutting the nerves where they entered the legs, that is, severing the long nerve fibers from their cell bodies in the spinal cord, wiped out the limb potentials almost entirely, but if I cut the spinal cord, leaving the peripheral nerves connected to their cell bodies, the limb potentials didn't change. It certainly looked as though there was a current being generated in the nerve cell bodies and traveling down the fibers. To have a current flow, you need a circuit. The current has to be made at one spot, passing through a conductor and eventually get back to the generator. We tend to forget that the 60 cycle alternating current in the wall socket isn't used up when we turn on a light, but merely coursing through it to the ground, through which eventually returns to the power station. Since my measurements were positive over collections of nerve cell bodies and increasingly negative out of along the nerve fibers, it seemed a good bet that current was being generated in cell bodies, especially since they contained all of the good stuff, the nucleus, the organelles, and metabolic components that, while the fibers were relatively uninteresting prolongations of the body, at the time, I suppose the circuit was completed by current going back toward the spine through the muscles. This was a good start, but it wasn't scientifically acceptable proof. For one thing, my guess about the return part of the circuit was disproved when I measured the limb muscles and found them polarized in the same direction as the surface potentials. For another thing, it had recently discovered that amphibian skin itself was polarized inside versus outside by ion differences, much like the nerve's membrane resting potentials, so that it was just barely possible that my readings had been caused by ionic discharges through the moist skin. If so, my evidence was literally all wet. Much of the uncertainty was due to the fact that I was measuring the outside of the animal and assuming that generators and conductors inside were making the pattern I found. I needed a way to relate inner currents to the outer potentials. This was before transistors and entirely replaced vacuum tubes. The tube's characteristics depended on a structure of the electrical field inside it, but to calculate the field parameters in advance without computers was a laborious task. So radio engineers often made an analog model. They built a large mock-up of the tube filled with a conducting solution. When current was applied to the model, the field could be mapped by measuring the voltage at various points in the solution. I decided to build a model salamander. I made an analog of the creature's nervous system out of copper wires. For the brain and nerve ganglia, I used blobs of solder. Each junction was thus a voltaic battery of two different metals, copper and the lead tin alloy of which the solder was made. Then I simply sandwiched this nervous system between two pieces of sponge rubber cut in the shape of a salamander and soaked the model in salt solution to approximate body fluids and serve as an electrolyte. The conducting solution that would enable the two metals to function as a battery. It worked. The readings were almost exactly the same as in the real salamander. This showed that the direct current inside could produce the potentials I was getting on the outside. My proposed system was really primitive part of the nervous system. It should be widely distributed, so I surveyed the whole animal kingdom. I tested flatworms, earthworms, fish, amphibians, reptiles, mammals, and humans. In each species, the potentials of the skin reflected the arrangement of the nervous system. In the worms and fish, there was only one area of positive potential, just as there was only one major nerve ganglion, the brain. In humans, the entire head and spinal region, with its massive concentration of neurons, was strongly positive. The three specific areas of greatest positive potential were the same as the salamander: the brain, the brachial plexus between the shoulder blades, and the lumbar enlargement at the base of the spinal cord. In all vertebrates, I also recorded a midline head potential that suggested a direct current that, like postulated by Gerard, flowing from back to front through the middle brain. It looked as though the current came from the reticular activating system, a network of cross-linked neurons that fanned out from the brainstem into higher centers and seemed to control the level of sleep or wakefulness and the focus of attention. At the same time, to see whether the current of injury and the surface potentials came from the same source, I made electrical measurements on the salamander limbs as they healed fractures. As mentioned in chapter 1, bone healing is the only kind of true regeneration common to all vertebrates. The limb currents behave like those accompanying the regrowth. A positive zone immediately formed around the break, although the rest of the limb retained at least part of its negative potential. Then, between the 5th and 10th days, the positive zone reversed its potential and became more strongly negative than the rest of the limb as the fracture began to heal. Next, I decided to follow up Burge's experiments of 2 decades before. I would produce various changes in the state of the nervous system and looked for concomitant changes in the electrical measurements. To do this right, I really needed a few $1,000s for an apparatus that could take readings from several electrodes simultaneously and record them side by side on a chart. My chances of getting this money seemed slim unless I could publish another paper fast. I decided to use the equipment I had for a simple measurement during one of the most profound changes in consciousness, anesthesia. Burge was right. The electrical responses were dramatic and incontrovertible. As each animal went under, its peripheral voltages dropped to zero, and very deep anesthesia, they reversed to some extent, the limbs and tail going positive. They reverted to normal just before the animal woke up. I had enough for a short paper, and I decided to try a journal on medical electronics recently started by the Institute of Radio Engineers. Although most of what they printed was safe and unremarkable, I'd found that engineers were often more open-minded than biologists, so I went for broke. I put in the whole hypothesis: analog nervous system, semiconducting currents, healing control, the works. The editor loved it and sent me an enthusiastic letter of acceptance, along with suggestions for further research. Best of all, I soon got another small grant approved and bought my multi-electrode chart recorder. Soon I had confirmed my anesthesia findings, and with the whole body monitoring set up, I also was able to correlate the entire pattern of surface voltages with animals' level of activity while not anesthetized. All right, guys, we're going to stop there. That's on page 100. So we'll pick up on page 100.